On to the main order of business, which is to present Eberhard Fetz with our 2024 NCM Distinguished Career Award. So Eb is an emeritus professor of physiology and biophysics at the University of Washington in Seattle. He received his undergraduate degree in physics from Rensselaer Polytech and then his PhD in physics from MIT. He then moved on to University of Washington for postdoc work and then apparently loved Seattle so much that he just never left. And he's been on the University of Washington faculty ever since. His research focus is on the neural control of limb movement in primates and mechanisms of neural computation. And in a series of pioneering studies, Eb developed operant conditioning protocol in which monkeys drove the needle of a meter, a now famous experiment using neural feedback, biofeedback, and showed that the neural activity could be used to drive an external advice, effectively establishing a precursor of current brain-machine interfaces. And in this context, Eb was featured in a relatively recent Economist article with the byline, "The Man Who Helped Launch Brain-Computer Interfaces." Eb's lab also pioneered the recording of spinal interneurons in behaving monkeys, and this is incredibly challenging and technically demanding work. And he demonstrated that spinal neurons share many of the same properties of cortical cells, which was really shocking at the time, including preparatory activity prior to instructed movements. He also investigated, again, in these incredibly challenging experiments, synaptic interactions between spinal and cortical neurons using in vivo intracellular recording. It's just incredible, and spike-triggered averaging. And then most recently, his lab has developed an implantable recurrent brain-computer interface that records the cortical activity of cells during free behavior and converts this activity to real-time stimulation of cortex, spinal cord, and muscles. And this so-called neural chip creates continuously operating artificial feedback to help the brain learn to incorporate into behavior and generate synaptic plasticity. And then finally, I should note or add that Eb has a long-standing interest, as many people know, in creative and artistic ways to represent the operations of the brain. So he's also been a professor in the Department of Digital Arts and Experimental Media while at the University of Washington. So with that, I'm going to move to the couple of PowerPoint slides I put together. And I was thinking, I said in my initial comments on the opening day that Eb was a sort of founding member of our society, so I wanted to go back into some of the old programs. And it wasn't very hard. I struck gold on the first thing I looked at. So this is the program from our first 1991 NCM at Marco Island, which few of us may remember. I was a postdoc back then. And session one, guess who organized session one of our first ever meeting? It was Eb. I just think that's amazing. So, yeah, I should go to the next one. Oh, I'm hitting the wrong key. There we go. So, neural network models of motor systems already looking to the future. Eb's talk today is aptly entitled "On the Neural Control of Movement." And I took the liberty of taking a couple provocative lines from his abstract, and we'll see what he tells us about. So you can read this, of course, but says trajectories of population activity in multi-dimensional neural space have led to the concept of lower dimensional manifolds, constraining dynamics, etc. And he says that such descriptions are conceptually seductive, but evade harder details about how neural computations of the brain causally generate volitional movements. So towards this end, he suggests combining operant conditioning with multi-unit recording and network models, and I think this will be the top part of at least what he'll tell us about. So with that, it is my sincere honor to present Eb with the 2024 Distinguished Career Award, and I will turn the podium over to Eb.
Well, thank you very much, Kathy, for this wonderful honor, and all the members and people in the society that decided on this honor, which I'm happy to accept on behalf of the many colleagues that have worked with me, that I've been lucky to work with through the years, that have generated a lot of interesting results, which I hope to show you some of. And so, as Kathy said, I attended some of the first meetings at Marco Island. So I realized, by the way, that this honor is sort of ironic in this context of my not being a member of the society for many years and not having gone to meetings for many years. And so the committee that decided on this, I really appreciate their going out on the limb and offering this to me anyway. So now the first meetings were held in Marco Island, down here in the tip of Florida. For those people who are maybe not so much aware. So I wanted to comment also that the society meeting has gotten much, much larger now compared to what it was at Marco Island. I think there was, 140 people in the first meeting, and now it's almost quadrupled. So I think it's to the benefit of the attendees, that it includes many younger people that are now much more encouraged to attend, and that's thanks to Lee Miller and other previous organizers of the meeting, and I think it's a major improvement. And so it also gives me an excuse to review a little bit of history, which might be of interest. So for those that are skeptical about my credentials, I want to just show you some evidence that I did actually attend these meetings here in Marco Island. And one of them is this picture, which was taken in Key West, where we flew with Alan Gibson, Mitch Glickstein, and John Stein, and they were the only ones who could drink, but I had to fly. So this was one of the excursions. Another excursion went up to Tampa. Fortunately, Marco Island had a nice airport where you could rent the Cessna. And here's Steve Promoter and Roger Lemon in one of these excursions. And so this illustrates that one of the points of the meeting is an opportunity to get together with friends and colleagues and also indulge in extracurricular excursions like this, including some diving. When Barry Peterson was the head and was an avid diver, the meeting used to be held at great dive spots. So anyway, this I offer as evidence that I am actually connected with this society, and would like to now ask this question: how do neurons in the brain generate movement? Okay, so we're all here to sort of probe that question. And as a movement, I would like to offer what Carl Lashley used to do for students before a talk. He would say, how do I do this? Right, how does my brain do this? And I like to think of that as sort of a koan, a Zen koan. And the question is, how does a brain do this? So the Zen answer, of course, is non-conceptual. I just do it. On the other hand, this whole meeting, your careers, everything is devoted to a conceptual wrestling with that issue. The Zen koan, of course, is designed to break through this conceptual impasse of between us and reality, but and hence this demonstration. But of course, the meeting and our approaches are all conceptual. And this week I had the revelation that our conceptual minds create all sorts of problems about neural control of movement that the brains go about trying to solve. And if we look back, these questions have been there since the beginning of the meeting. And I'm going to show you some examples about the fact that we're still investigating some of the same issues with much more sophisticated instrumentation and technologies and approaches, but basically the ideas are still there. the challenges are still there in terms of understanding how the brain controls movement. So here, to give the illusion of organization, I'm going to show you an outline of what I plan to discuss. One is to just start with the early concerns about what do neurons actually correlate with? What do they code, in other words? This was not known until neurons began to be recorded. And I'm going to go through some of these early experiments which may be new to the young people, but may also be interesting to the old-timers as to how the same questions are still an issue today. So then I'm going to move to premotor neurons that facilitate muscle activity. So this is a subset of neurons whose connections to motor neurons allows them to be identified by spike-triggered averaging. And they have a sort of special relation to movement and muscles. And then switch to the question of what can operant conditioning of neural activity do to clarify any of this, or perhaps muddy it up, and consider what neural network models can do to elucidate the underlying mechanisms, and then address the issue of population mechanisms, what they can tell us, and actually a much briefer comment on future directions than Kathy would have led you to believe. And finally, hopefully there's time for something completely different, which you'll see later. So starting now with what do neurons correlate with? We're talking about primarily neurons in the motor cortex, many of which have descending connections to the spinal cord. And before the advent of single unit recording in awake monkeys, this was an open question. What do they actually do? We knew about where they go, but what do they do? So one of the earliest experiments of a pioneer in the field of chronic unit recording, Ed Everts, designed this experiment to answer the question whether these neurons were in the motor cortex, not just neurons, but pyramidal tract neurons, were more strongly related to where the monkey moved or the muscle force that was created. So he designed a simple experiment in which these two parameters were dissociated, or could be dissociated, with the monkey moving a handle against different weights. And the answer to the question was that most of the pyramidal tract neurons were modulated more strongly with force than displacement. So it was really, that makes sense, because motor cortex descending command should be driving muscles, so force is it. But nothing's simple long. So, Apostolos Georgopoulos came along and had a monkey move in different directions and looked at the activity that's associated with these movements, plotted the firing rates of cells, and demonstrated that many motor cortex cells have this sort of cosine tuning with maximum activity in a preferred direction. And you could actually recreate the direction of movement by adding up the cell firings times their preferred directions, and that was quite intriguing demonstration of the coding of displacement. Then, of course, John Kalaska, let's see if I can, came and showed it was actually both, that neurons in the motor cortex were modulated both by direction of movement and force, and in his experiments, the monkey moved in these different directions against loads that could be spatially changed. And there were relations of static hold, which are these big circles, and also direction of movement, which are these angled plots. And so there was a relation between displacement, direction, and force. So it got a little bit more interesting when, so you have to appreciate these were cells that were selected out of a large group of cells which might not have been so clean as this. But one of the early experiments showed that population activity is even more capable of showing the correlation with movement parameters. And this is the first study, really, as far as I could tell, of demonstrating the power of population coding. And Humphrey, Schmidt, and Thompson did a similar experiment to Everett's and correlated, in this particular example, the force with the weighted sum of firing rates of 5 cells. So these are the firing rates. Sorry, you probably can't see it too well. But just to demonstrate how populations are better, you can superimpose the force trace here with the firing rate of the most correlated cell. And it's okay, but if you add additional cells, you get an improvement in this correlation. So this is only 5 cells. Of course, each one is added with the coefficient that optimizes the correlation. But nevertheless, the point is that if you have a larger population, you can extract this parameter with a simple linear decoder, this weighted sum. And the correlation gets better with the number of cells included. So then the other interesting point in these experiments is that it's not just force, but also displacement, velocity, rate of change of force that could more or less be extracted, as it were, with this simple formula, from the firing rates of these 5 cortical neurons. So in principle then, you could get pretty good, accurate correlation of cell activity with a particular parameter of movement with large enough number of cells. And the number of cells included in these estimates went up with Miguel Nicolelis's multi-channel recordings, and Jose Carmina did the study that I like because it shows several interesting things. So this is a monkey moving a cursor into a target, and it can also generate force. And number one, and these curves represent the degree to which this formula can match these parameters as a function of the number of neurons included in the sum. So the fact is that they could match position, velocity, gripping force with the same population. Of course, the secret here, the key is that the coefficients were optimized for each situation, but nevertheless, implicitly, there is the ability to extract these parameters. So number one, different parameters. Number two, numbers of cells improved the thing. And number three, these different curves are from different cortical areas. So the red one is motor cortex, and these others are like S1, posterior parietal, supplementary motor area, medial dorsal, premotor dorsal rather. And so another significant discovery here, or demonstration, was that cells in many areas of the brain, across the brain, can actually represent these different parameters. So it's not like primary motor cortex is the only place where you can find cells related to force. So this distributed, so these again are phenomena that are very well known to most of you, I'm sure, and have been driven home with much more precision and much larger populations of cells, but nevertheless were anticipated by these experiments many years ago. So just to summarize some points about coding before moving on. So cells can be found that covary with any number of different parameters, and many movement parameters can be extracted from the same population of cells. The degree of accuracy increases with the number of non-redundant units. Non-redundant meaning that, they are adding something new to reducing the remaining error. And cells with similar properties are widely distributed in different cortical regions. So does this mathematical extraction actually demonstrate anything about biological functional coding, or is this a mathematical exercise that is possible, but it's not really the way neurons compute? The way neurons compute includes their connections and their particular contributions to the movement, and this mathematical extraction is nice, but not necessarily proving anything about the underlying coding, underlying mechanisms of motor control. So what we did was look at, that led us to look at cells that had actual output effects on muscle activity to nail down what the relationship would be one synapse back from the muscle, right? These are cells that have monosynaptic connections to motor neurons, and they are identified by spike-triggered averages of EMG. So this is work with Polcini that used the action potentials of cells to trigger averages of rectified EMG activity, while the monkey just did the simple alternate flexion extension task. And for certain cells, one saw this increase in the probability of the muscle activity called the post-spike facilitation that suggested that this would be a cell whose axon goes down and actually connects with the motor neuron. So this is a special type of cell that has a particular identifiable connection to motor neurons. And one can see already that there are several different muscles that show this facilitation. And this diagram summarizes the sort of divergent connections that these cells have to multiple synergistic muscles, and it also includes connections to inhibitory connections to antagonists of their facilitated target muscles. So this represents the post-spike suppression of the cell, of the EMG in antagonists of the muscle. So you have these CM cells sort of coding or distributed in this way, and the question is, what is their activity? The activity we monitored or documented in terms of the activity in relation to the simple alternate flexion extension. So we have a phasic and a tonic component of the movement, and in this context, the cells these CM cells had phasic tonic mostly, or tonic. But then there were some peculiar, interesting outliers. 10% of them had this phasic ramp profile. So the point here is that the activity doesn't necessarily mirror the activity of the muscles. The cell can accentuate the input to the onset of the movement, and then also this ramp activity can help overcome the tendency of motor units to adapt. So there are specializations, and the activity doesn't necessarily just simply co-vary with the target muscle activity, even the target muscle activity. So this same strategy was applied to other premotor cells, such as spinal interneurons, and I'm just going to show you one example of an interesting inhibitory premotor interneuron that is shown here in terms of its post-spike suppression of activity of this flexor carpi radialis muscle. But the curious thing, non-intuitive, is that it co-varies with the muscle that it inhibits. Okay, so it's not always a simple matter of having excitatory connections to co-activated muscles, but there could also be sort of inhibitory connections. And these look strange, but they're the 1A inhibitory interneuron that was identified in acute experiments has this sort of predicted profile. And it's also, this cell is also active in the opposite direction. So to basically summarize or compare the connectivity and the activity of cortical versus spinal premotor neurons, this little summary shows that CM cells typically produce post-spike facilitations in multiple muscles and can produce inhibition and antagonists of the ones that they facilitate, whereas the premotor interneurons had more restricted target fields and included cells with monosynaptic inhibitory connections. And the activity, which is the other side of the functional story, is that in terms of this flexion extension ramp and hold task, CM cells had strict activation in one or the other, but not both, whereas the spinal interneurons had typically showed much more activity and often in both directions of force. So you can summarize that as interpreting the fact that the CM cell represents functional synergies of muscle contraction much more than the interneurons. So these sort of studies were done with different premotor cells. I showed you the CM cells and the premotor interneurons, but Polcini investigated rubromotoronal cells. We looked at dorsal root ganglion afferents that produce post-spike effects, and then there's a population of unidentified interneurons. And this plot, shows the proportion of cells showing these different response patterns in relation to the simple ramp and hold movement. So they were all recorded under the same conditions, and all these populations had a phasic ramp, phasic tonic discharge. They had cells with tonic discharge and cells with phasic discharge, but in different proportions. So what that tells you is that all of these cells are sort of co-activated with the movement in this way. Motor units are shown here individually as having also similar sort of patterns. And once again, there's these outliers that are intriguing because they represent functional involvement that one can identify only if you know that this is the firing pattern and the cell actually affects motor neurons in proportion to that firing pattern. The other interesting category that Polcini discovered is these unmodulated rubromotoronal cells that they became active with movement, but they weren't actually modulated with the movement. So to summarize then, the premotor neurons, they have, first of all, direct correlational output effects on motor neurons, and their activity can differ from the activity profile of target muscles in this ramp and hold movement. And then, intriguingly, some are unmodulated. I'm coming back to this point, that's why I'm repeating it. And some spinal interneurons are counterintuitively co-activated with muscles that they inhibit. So this is, it doesn't all fall into, even for this population of premotor neurons, it doesn't fall into simple categories. So now I'm going to take a little detour and prepare you for a change, and that has to do with the fact that the relations between cells and muscles can be investigated by directly operantly conditioning the cell and muscle activity. So this is something that I started as a postdoc in Seattle, training monkeys to control motor cortex cells. So here's a precentral cell, which the monkey is rewarded for firing at higher rates, and the rate was measured by this activity integrator. And when it reached a certain threshold, the monkey got his applesauce, and the monkey also saw in the form of the meter how close his activity was getting to the reinforcement level. So an interesting point about these experiments is that the monkey quickly learned that there's something he can do to drive that meter to the reinforcement level, and we could change the requirements for driving it to reinforcement rapidly and quickly within the experiment. So here's just a simple cell recording and reinforcement again in the study with Marianne Baker. This is preconditioning, then the feedback and applesauce was delivered, and the monkey quickly raised the firing rate, and we raised the threshold so that he increased the rates for as long as it paid off, and then the rates went down again. So the volitional control of these cells was really remarkably specific. Here's two neighboring cells with different wave shapes, and the firing rate of this large unit and the small unit are plotted in 1 minute averages here. And here the small unit drove the meter to reinforcement. Within a minute, he was driving the activity up, time out, back to baseline. Then the large unit drove the meter to start. This is the first time the monkey saw any of this, but just to give you a sense of the fact that they can very quickly modulate this activity volitionally. He increased the firing rate of the large cell, and then during timeout, they went back to baseline, and to then ask whether the monkey could actually reduce the cell's activity. This period just required that he shut this large cell off in order to let a multi-vibrator make the meter drift to reinforcement, and after a couple minutes of trying what worked before, there's a reduction in the cell activity. So this shows the two things: bidirectional activity, volitionally controlled, and independent control of two neighboring cells. And interesting enough, these two cells both responded to the same sensory input, which was passive joint movement of the knee. So the point is that there's evidence here of very specific volitional control onto these cells when reinforced. So in general, the basic biofeedback paradigm involved recording some reinforced activity in the brain, making it explicit through feedback, which this volitional controller, which is of course the rest of the brain, in a significant black box here, can then volitionally control the reinforced activity. And the other elephant in the room here is the correlated activity, what's going on with the reinforced activity. And in particular for motor cortex, the correlated activity could be muscle activity. So is he driving muscles, or do muscles activate the cell? So we decided to include the muscles in the reinforcement contingency by, in this study with Dom Finocchio, the idea was to see whether the cortical cell could be shown to be active with one or more of the arm muscles, so that would be biceps, triceps, and wrist flexor, wrist extensor, EMG. They were all recorded, and a weighted sum of their activity was now driving this integrator to threshold, and the monkey saw the meter that showed him how close he was getting. And the weighted sum could be such that activity in some muscles that we wanted him to suppress would have negative coefficients, and activity that we wanted him to create would have positive coefficient. So tweaking these, the polarity and gain of these weights produced the ability to ask a monkey to demonstrate that he could generate these different activities. So here's the summary of the firing rate of the cell in the histogram at the bottom, and the profile of EMG activity in the top of these are the four muscles. This is the flexor was driving the meter to threshold. The minus means that if he co-activated these muscles, that would prevent reinforcement. And 0 means the unit was not part of the contingency. And in this case, he co-activated the cell with the wrist flexor. But over here, with the biceps being reinforced, he co-activated the unit even more strongly, and not so much with either of the extensor muscles. So this already showed the possibility of cells being co-activated with different muscles, and in the paper there's also a demonstration of different types of relationships with these different muscles. But an interesting point here is that even though this particular cell was co-activated with the biceps, when the biceps burst was reinforced, and when he actively moved the elbow, when he flexed the elbow, and also when he was reinforced here for increasing unit activity without any contingency on the muscles, he co-activated the muscles. But surprisingly, when he was required to suppress the muscle and activate the unit, he went into that pattern as quickly as any of the others. But it did take a while, and we, of course, probed for the opposite dissociation, namely biceps activity without unit activity. He did increase the biceps activity a lot and reduced the unit activity, but didn't suppress it entirely. But that was after 8 hours of reinforcement and applesauce, and it wasn't clear whether that was explanation of inability to completely dissociate in the other direction was a behavioral limit or whether it was a physiological limit. So we revisited this question in the meantime with a more interesting group of cells, which is namely the CM cells. And this is unpublished work with Yukio Nishimura and Ryan Eaton and Steve Perlmutter, where a monkey controlled a cursor in the X and Y direction with CM cell activity in the Y direction and EMG activity in the X direction. And we could present the target in various portions of this space. And clearly when the target was presented for co-activation or co-suppression here, there were a lot of these dots represent the averages in a certain window, but the co-activation and co-suppression was quite clear. But you could also see that when the monkey was rewarded for increasing the cortical cell and suppressing the muscle, he readily got into this area of the space, and with muscle activity, with the cursor down here, requiring muscle contraction and the cortical motoronal cell suppression, he also dissociated in the opposite direction. So that shows that even these directly connected elements, cells and muscles, can be dissociated with direct operant conditioning of that pattern. In other words, it's not a natural thing, but if you require it, then the monkey can produce it. So this is just the average the profiles of activity for the CM cell and the target muscle when the monkey was required to increase cell activity and decrease muscle activity, and the dissociation in the opposite direction showing cell suppression and muscle activation. So this shows an unexpected degree of control over two elements that are synaptically connected, probably. Again, the point is that operant conditioning of that activity can reveal that flexibility. So a summary of operant conditioning. we can use direct operant conditioning to explore possible volitional activations beyond the relationships that appear in normal motor behavior or task performance. And motor cortex cells that consistently covary with muscles can be volitionally dissociated with operant conditioning, and even activity of CM cells can be dissociated from activity of their target muscles in both directions. So operant conditioning, I would suggest, is a fairly powerful way of exploring new levels of activation that are not going to appear simply from trained behaviors. Okay, so I'm now going to do an abrupt switch to the next topic of neural network models, because I think that's part of the story I'm going to tell. And that has to do with the fact that neural network models can provide complete solutions for populations of cells. In the example I'm going to show you, the task is the step tracking task, where the monkey sees a cue that tells him to either flex or extend, and then he activates the flexors or extensors going alternately. So with Larry Shoup, our brilliant programmer, we did this dynamic recurrent neural network model that used these signals as inputs to the network, flexion, extension signals, 4 of them, 4 variations of them, tonic and phasic. And then these were fed to excitatory and inhibitory hidden units, and the network had to generate the 4 types of motor unit patterns for flexor and extensor motor units. And the technique was a backprop technique of changing the weights in a way to reduce the error, that is the difference between what the network was producing and what the target was. That determined the change in the weights, and these things converged relatively robustly depending on what the input was. So the input could be just simply the phasic inputs or the tonic inputs. I'm just showing you the complete set, but it doesn't mean that that was the only thing that these networks could learn to do. So just the phasic inputs could require the network to say, integrate that and then have a phasic hidden unit that produced this tonic, helped produce the tonic output. So just as an example, and by the way, yeah, this is an example that was presented at the very first session, at the very first meeting, and was criticized by John Kalaska as being ridiculously trivial, which I agree with. I'm just showing it to you to make some points about these networks. So this is the complete representation of the network in terms of the connectivity between units and their activity. So if you look down here, you see the profiles of the inputs, the excitatory units, inhibitory units, and outputs. And the strength of the weight of the positive in black is coded by the size of the square. So this is what I mean by having a complete solution. You've got it all. You've got the connections, and you've got the activity. And you can start analyzing, how these patterns are produced. So for example, this happens to be a phasic flexion output motor unit, okay? You look down here, and you see it's getting strong input from this tonic hidden unit, which is interesting, but you go down further and you see it also gets strong input from a tonic inhibitory unit, which starts a little bit later. So this is an example of 2 inputs that don't match the output, but in combination contribute to the output. This is just a very simple example, but these network solutions are full of these sort of examples of how hidden units that have complicated relation to the output, nevertheless, in combinations with other complicated hidden units, can contribute to the output. So these are really great to work with because you can very quickly not just analyze it, but also manipulate it. You can make lesions, see what happens if you cut out this unit or stimulate. You can produce some activation in these cells. You can do virtual experiments with these networks that provide lots of insights, and they're fast. You can just run the simulation really fast, and it doesn't require recording in a monkey to do this. So I'm sold on neural networks as a powerful way to analyze mechanisms of control. This is, as John Kalaska said, ridiculously simple, but this is what we're seeing today in AI, for example. AI is based on training artificial networks on examples of behavior. That's the other thing about these that I didn't emphasize enough, is that these connections are obtained from examples of the behavior. You don't have to pre-wire things according to rules and preconceptions about how things should work. You can just let the network optimize its connections to perform examples of the behavior. So I think it's great. Okay, so here's probably going to repeat the same question. So the summary of neural networks is dynamic recurrent neural network models can provide complete solutions for spatiotemporal input-output behavior. And the weights can be derived from examples of the behavior, just like today's generative AI, which is doing it on steroids, of course, but it's the same phenomenon. Well, I mean, pretty much. The activity of the hidden units can differ significantly from the outputs that they contribute to. And so one conclusion, for example, is that these networks can operate without requiring any explicit coding of the output in the hidden units. So let's see what's next. Oh yeah, so all of this is leading up to this critique that I wrote way back in '92 about neural coding. At that point, coding was pretty much the name of the game, how to find out what the cells are about. You do these experiments. And in this critique, I pointed out that in these recording experiments, you actually find neurons that can be categorized in terms of three types. One type is interpretable. It correlates with the variable that you're interested in showing is being coded, and those are the ones that get into the figures, and those are the ones that get discussed, and those are the ones you saw. But even larger population of cells are modulated, but their activity is just not interpretable in terms of a direct relationship to what you conceive of as a variable that needs to be coded. Some are paradoxical. They are counterintuitive. They don't even seem to make sense. And even larger population of cells are simply unmodulated. All right, so I think you see where I'm going here. The fact is that all three types of units contribute to the output in the example, two examples that I showed where we know not just the activity, but the connectivity, okay? And those two examples are premotor populations of monkeys that we know the output goes to muscles, but they have all three types of cells. And in neural network simulations, where we can trace the connections to the output, we have all three types of hidden units. So what that says is that rather than try to analyze the control of movement, let's say, in terms of searching for coding, explicit coding, this distracts us from recognizing a radically different mechanism of neural control. That was the conclusion of this paper. So what did I have in mind? I had something in mind which I'll tell you about, but interestingly enough, one of the radically different mechanisms was cited by Shenoy and Churchland and others who were proposing this dynamical systems approach. So they quoted this statement: the search for neural correlates of motor parameters may actually distract us from recognizing the operation of radically different neural mechanisms of sensory motor control. And they said that 20 years earlier I had argued for the dynamical systems perspective. Well, I just am here to set the record straight that I did not have that in mind. I had something else in mind, which I'll tell you about. But as you know already from what Kathy showed from my abstract, I'm somewhat unappreciative of what this dynamical systems approach can tell us. So as you are surely aware, the approach is to add up all the activity of a cell and represent the state of the system as a point in multidimensional state space where you have these coordinates that represent the neural activity. And when that's done, one sees that the dynamics of this population point in reproducing behaviors also reproduces certain pathways, and that's all interesting. Here's another example of taking population activity of many different cells that have a variety of activity, and we already know that the cells have a variety of activity, but if you boil them down to one parameter, that's the sum of them, and watch what happens during a task, you can see that the pathways are constrained to a particular subset of all of the space, which is sometimes called a manifold on which it operates. And one can describe the dynamics within that manifold, which is in a cartoon form shown as a plane here, in terms of other parameters that describe the dynamics, and in fact the units can be shown or can be extracted from these parameters by weighted sums of activity. And this is all very nice, except it doesn't really explain anything. It describes very well what happens to the overall population, but it doesn't really even give you a mechanism for a handle on how the neurons are doing the computation. In order to do that, you need to identify the neurons and look at their connections and then try to piece this together. That's frustrating, it's got complications, but if we're talking about neural computation mechanisms, that's information is sort of critical to it, I think. But I'm sure I'm missing something because there's a lot of enthusiasm about this model. The other problem, I think, I'm not kidding, I think I've oversimplified here because I've missed something that the enthusiasts will tell me about. The other interesting point is that this manifold theory, that things are restricted to a manifold, is just begging to be tested by operant conditioning. So here, let me go back to this situation of a CM cell and a motor units in the target muscle, which normally coactivate. And you would think they're on the same manifold, right? But if you give the monkey applesauce for showing that he can get off that manifold, it happens quite readily. So another way to put it is that I think one can explore the restrictions in population activity in manifolds with operant conditioning. I want to show you one other example which I like a lot, and that is this study of Carmina and Costa on volitionally rewarding rats for controlling independently activity of visual cortex cells. Okay, now we're not in motor system anymore. This is visual cortex, and they just simply took some cells, ensemble 1, which drove the sum of activity toward the reward tone. In this case, the rats were getting auditory feedback since they couldn't do visual feedback. And another ensemble of neurons drove the sum of these populations in the other direction. And lo and behold, the rats could get their reward by driving one ensemble up and the other down, or vice versa, volitionally. Okay, so this volitional system got its tentacles even out to sensory cortex. So what's possible to demonstrate with this operant conditioning is that there is a volitional control of cells even in primary sensory areas like visual cortex. And it didn't depend whether the rats were doing this in the light or the dark. So there was a slight difference, but nevertheless, you could do it in either context. So it was not a simple trick of moving the eyeballs in the right way to drive a cell, as you might expect, but there is some central input. So I think the manifold area of research is definitely ripe for testing in terms of what one can directly operantly condition the animal to demonstrate how restricted the activity actually is to those. So then let me just summarize the dynamical systems. It describes, well, I think I won't bother you with this. Trajectories occupy a subset of the entire space and repeat with repeated movements. Not entirely surprising, actually. And such descriptive exercises totally ignore the activity and connectivity of individual neurons. So they've just eliminated the possibility of getting a functional understanding of how neural computation operates. It's a description, it's not an explanation. Okay, I'm beating a dead horse here. But operant conditioning can be used to probe this the fact that neurons can be dissociated from their typical manifolds. Okay, so dynamical systems was not the radical direction other than coding that I had proposed in that review article. So another population mechanism that's radically different from explicit coding involves holographic mechanisms, okay? So get ready for a sudden transition, another sudden transition, to explain what I mean by holographic mechanisms. And the best way to illustrate those mechanisms is by actually looking at a hologram and seeing what that means. It's a model, it's a metaphor for what's going on in neural networks. So don't get upset if it doesn't look like anything having to do with neurons. So what holography does is represent something in a non-literal form. It's not like a photograph. Instead, the photographic plate is recording the coincidence or the superposition of two waves. There's a reference beam. This requires laser light, which is very coherent, and you combine the original laser light, which is the reference beam, with the light that's reflected from an object. So the object has produced changes in phase, and the photographic plate essentially records where the two waveforms superimpose positively or cancel negatively. So it's now recording a phase information. And after the plate is this is what happens to your brain, sorry, is developed. What you have is a hologram which represents this object in a unique way. It basically requires a reconstruction of the waveform by illuminating the hologram by this original reference beam. And what that does is the hologram is sort of like a diffraction pattern, a diffraction grating rather, that reconstructs a waveform that represents the object. That's the virtual object here, okay? And so the information is there about what the object looks like, but it's coded, if you will, in a way that doesn't represent the object at all. It just represents the relationship in phase between these two waveforms. So here's an example of the fact that spots on a holographic plate have no relation to the stored image. So this is the holographic plate, and this is what you see if you illuminate it with the right reference beam. And as you can see, this has no resemblance to the image. Instead, what these spots represent is what has to be there in order to constructively interfere with the waveforms that are diffracted by the rest of the plate. Okay? So basically, this is an example, simple example of a non-literal representation of 3D information. So the intriguing thing here is that there are some similarities to the way neural networks work. So this was first identified here. Well, one of the first people to identify it was Ottmar West, who did this thesis in Darmstadt, showing that memory could be seen as a holographic mechanism. And for example, when you look at cells, they're firing when they get coincident input. It's like the positive superposition of the waves, and they fire when that input exceeds threshold for firing, and it's a distributed system, and it's just the natural way that neural networks work. So if you want to list the similarities, it's intriguing, but I'm not basically pushing this as the only thing going on. But the fact is that holographic mechanisms and neural networks have similar properties. Number one, this non-literal representation in the medium. Second, there are inherent associative mechanisms, which I didn't show you, but the holograms can reconstruct other parts of the hologram just from part of the input. The brain does this association automatically. There's a gradual degradation of the image with lesions. So if you take half the holographic plate away, the remainder can still reproduce the whole image. It's noisier, but it's there. And then you have multiplex storage in the same medium automatically, because it depends on what sort of reference beams you use and so forth. So those are intriguing similarities in the properties of holograms and neural networks. And in fact, one can argue that the mechanism for this has similarities in the sense that constructive interference of these optical waves is analogous to coincident input that activates successive neurons. And these neural network models implement holographic mechanisms. They automatically do. Not just the models, but probably networks in the brain. But it's not that simple, obviously. It's not like an optical hologram, because the brain operates dynamically, for one thing. It's not just a static image. And it does exploit representations. You do have coding. You have, the brain somehow figures out how to optimize its representations by having some cells that have explicit coding. Face cells and place cells are definitely there. They're not in that model, that are the optical model, but the brain has figured out a way to include that. But the part of it that involves this distributed representation is still basically analogous to these mechanisms. And so that's what that review sort of hinted at. And I still like the idea, but it needs to be obviously confirmed how much that model works. All right, let's see. Okay, so here we go, finally, future directions. Oh my God. So the mechanisms that control movement, I would propose, could be further investigated using neural network models that simulate the behavioral populations of interconnected units. So what do I mean? Why is that such a promising direction? Number one, such models can incorporate the known connections, activities, and learning rules. So we know something about how the brain is connected and so forth. And you can put that into all those into the model as constraints. They can be trained on examples of the behavior or through interactions with the environment. So this is what AI is doing today. It's being trained on examples of behavior. It's generating internal representations. And when AI gets embodied in robots, we're going to see AI that has a sense of self and interacts with the world the way we do. It's just going to happen. And it's all done with the basic neural mechanism, the capacities of neural networks to process this sort of information. And the example that I showed you of providing a complete set of all neural activities and connections is another big advantage of these models, because they can show you how the components are interacting, and you can see it all laid out. It's not like a monkey where you put electrodes in and you see part of the thing and you don't see the rest of the system. You sort of wave your hands and say, well, the rest of it is being handled by something else. It's inherently incomplete, whereas the networks are complete. And another nice thing is that they can be quickly tested for the effects of lesions and stimulation and novel inputs. It's very seductive. You can basically play with these models and see what the outputs, how the outputs change in the face of these sort of manipulations. So I think it's fairly powerful. And they can be used to test new hypotheses about how mechanisms operate. Okay, I think that's the point at which I want to thank all of these people throughout my career that have made the career possible. It's been a fantastic trip. And point out that a lot of the papers that have come out of all of the papers that have come out of the lab are available on our website. So some of them I stupidly published in fairly obscure places, but they're there. You can get a PDF. And then thanks to all of these granting agencies. So that's really the main talk. But if you give me another couple of minutes, I'll show you this final bit of, this is more like, we've had the meal, now here comes a dessert. So the fact is that now I've gotten very interested in using, in ways to communicate about the brain in nonverbal ways. I mean, here we're communicating among colleagues that sort of understand these concepts, but there's a whole world out there that is not trained in neuroscience. And I think that art can be a useful way to communicate how the brain works. So I just want to show you two examples. One is this collage, which is rendered on this white portion of the image is actually a mirror that reflects reality. And this figure is emerging from these complicated neural networks and representations of resonance and interactions and so forth, and discovering the reality out here. So this figure, as many of you probably recognize, is taken from this Flammarion woodcut in which there's someone who's breaking through the sky, through a barrier between the world behind him and the celestial mechanics out here. And so, in a sense, this is a metaphor. This image here is presented as a metaphor of the brain developing a self that explores reality. So that's one example. Now the second one, only 2 minutes more. The last one is actually a little video that was designed to communicate to anyone who watches it that it's all done with neurons, that what's going on while you're watching is represented by neural activity in your brain, okay? And the easiest way to communicate that message is actually to say it. So here's the thing. I hope the audio works. It's all done with neurons.
Okay, welcome to this little rap on the operation of your brain and its relation to experience. So let's begin by considering what's happening right now. Namely, you're watching this video of my talking head. However, this straightforward mental experience is actually being generated by neural activity going on in your brain. The activity of these neurons consists of pulsatile events, which, if amplified, would sound like little pops, like the punctate events you're now hearing and seeing. Different neurons are activated by different features, like brightness, edges, sounds, mental events, and so on. So consider the fact that everything that you are perceiving at this moment is actually generated through such neural activity. And moreover, this is actually occurring in your brain right now, in this instant. But your perception doesn't feel like the underlying neural activity at all. It just feels like you watching this video. Nevertheless, these clicks and flashes that represent neural activity are also triggering neural responses in your brain right now, which you are experiencing from the inside. So the main point of this piece is simply to make you aware of the fact that it's all done with neurons.
Okay, welcome to this little wrap. Okay, that's it. Thanks for your attention.
So thank you very much, Ed, for a very inspiring and thought-provoking talk, and really enjoyed
Crazy.
bringing the art in at the end.